Hello, everyone, and welcome back to this introductory lecture series for the Data Science Challenge. And this lecture is going to cover technology. So in the previous lecture, you saw this introductory material on T cells and how they fight cancer and become exhausted. And so what we really need are technologies to actually measure the T cells, characterize what they're doing, and learn how we can change those states. And that's really where the technology comes in this lecture. What we're really going to be focusing on to begin with is measuring gene expression in single cells. And so this is broken down into sort of three areas. One is revisiting gene expression programs, then talking about how we're going to measure those gene expression programs in single cells, and then how we're going to do perturbations to cells and measure gene expression programs in those cells. So gene expression programs. So to remind you all, genes are the fundamental units of heredity in all organisms, and they're passed down to us from our parents. And genes contain information that specifies all our physical traits, such as our height or our eye color, and also biological traits, such as how we respond to different diseases. And genes are written in DNA and using this 4-base code of adenine, thymine, cytosine, and guanine, or A, T, C, and G. So in each of our cells, genetic information flows from DNA to RNA to protein. So remember, our genes are encoded in DNA, and then this DNA can be copied or transcribed into an RNA molecule, and then that RNA molecule can be translated into a protein. And it's these proteins that are really giving the cells the different functional capacities that they have across these different tissue types. And so the genes and which genes are expressed are controlled by gene regulation. And gene regulation is going to lead to different genes and proteins being expressed, creating this functional diversity we see across the cells. And in gene regulation, we have gene expression programs. And these programs are groups of genes that are expressed together or turned on together during a biological process. So when a biological process gets turned on, RNA and protein need to be made. So you need more of an RNA and more of a protein in order to carry out that process. And so that's this idea of a gene being turned on, an RNA being made, and protein being translated. So by measuring the levels of RNA, we can learn what genes have been turned on or off in a cell, and that tells us what the cell is really up to. So to recap, each cell type and state is encoded by a different gene expression program, different sets of genes, and by measuring the levels of the RNA, we can learn what genes have been turned on and are defining that program. So now let's connect what we learned about gene expression programs to the T cell states that we previously discussed in the first introductory lecture. So to remind you, the T cell states in cancer are going from this progenitor state to this effector state, and finally this terminal exhausted state. And these states differ in their ability to kill cancer cells, where the progenitor states and the effector states are strong at killing tumor, whereas the terminally exhausted state is very weak at killing tumors. And this ability to kill tumors is really defined by the gene expression programs in those cell states. So each state has a different gene expression program. And now let's just look at a few of the genes that define those gene expression programs for each T cell state. So for instance, in the progenitor T cells, we see very high expression of this gene TCF7. And we know that TCF7 is important for maintaining the progenitor T cell state. So when we see high expression of TCF7 in a cell, we know that that gene expression program is really defining or kind of indicating to us that this is a progenitor T cell. Likewise, in the effector T cells, we see high levels of expression of PRF1. This is a gene that's important for forming pores on target cancer cells that lead to the death of the cancer cell. So that's a gene that we already know is important for the effector kind of T cell killing activity. So when we see high expression of PRF1, high expression meaning high numbers of RNA molecules, we know that that T cell is in the effector state. And then finally, we have the terminally exhausted T cell state, where we have high expression of the gene PDCD1. Now, PDCD1, we've actually seen before in the previous lecture. PDCD1 encodes the protein PD1, and this is a target of immune checkpoint blockade. And the PDCD1 gene that encodes PD1, it gets engaged, and it leads to the T cell killing activity being inhibited. And so the T cells lose their ability to kill target cancer cells. So when we see a T cell expressing the gene PD-CD1 at high levels, we know that that's likely a T cell that's in the terminal exhausted state. So these are just a few of the genes that define the gene expression programs for each of the T cell states. However, in reality, gene expression programs are defined by tens to hundreds of genes, not just 2 genes alone. So we really need technologies where we can measure the expression of genes, where we can look at how much RNA is being made for each gene and quantitatively measure that. And we can use those measurements to exhaustively define the gene expression programs for each of the T cell states. So we'll talk about what those technologies are. So that's the first problem we want to discuss. And then the second problem is once we have a good understanding of the gene expression programs in the T cell states, we would really like to perturb these cells. And by perturb, I mean apply a genetic change to this T cell such that we shift the distribution of T cell states to favor progenitor and effector states. However, there's many genetic perturbations that one could make in T cells. So in our genomes, there are 20,000 genes, roughly. And in theory, then you could make 20,000 perturbations. However, that's way too many to characterize experimentally in the lab. So we need to think of how we tackle such a problem.